the intent of this talk is to give an answer to this question. So the framework that I'm thinking for this question is, I think of calculus as being the natural way in which to express physics. So by physics, I guess I mean Newtonian physics or electromagnetism and things like that. And it's not, immediately obvious what this would correspond to for computational biology. So I'm going to give an answer to this question, which I think is the right answer, but you probably don't, and I hope to hear from you. And in doing it, the way I'm going to do it is by talking about what it is that we do here in my group. So we develop mathematical and computational tools for model-based statistical inference on continuous and discrete mathematical objects, motivated by molecular sequence analysis of microbes in the immune system. So this is going to be a bit of an outline for the talk. So we're going to first talk about model-based statistical inference. We're going to talk about what it means to do this on discrete mathematical objects. And then we're going to talk about mostly immunology after that. So I do hope that people ask questions if you have them. I think I'll be able to see your hands if you lift them, but if not, Jeremy has promised to keep me in line. Okay, so
I'm gonna pass around the microphone so that the two potential moderators will be on each side of the room.
Great. Okay, so what is model-based statistical inference? So I would say modern technology in general, and biological technology in particular, gives us the ability to observe things in great detail. But that's not the same as understanding them. And in particular, I think that to really understand something, you need to simplify it and abstract it. So here we have somewhat good reconstruction of, or the segmentation of this picture, this rather complicated picture over here, into different parts that the computer thinks are different. So it's sort of done a good job of identifying that there's a person here. It hasn't done a perfect job. Of course, it's sort of identified part of this building as being part of this person as well. But I would argue that this is a good first step towards understanding what this picture is about. So what sorts of abstractions do we have at our disposal? But what can we use? Well, starting very basic, we have, oh wow, okay, this is interesting. Well, we have three. We have the number 3. So that is really all this slide's supposed to show. So I'm sorry that you are missing part of the upper part of the 3, but I'll have to imagine that you can understand that there is an upper part of the 3. So 3 is an abstraction. It's not one that we usually think of as being an abstraction, but it really is. Like you can have 3 coins or 3 llamas or 3 baseball tickets in your pocket, but you can't have just 3. So it's an abstraction that we're fairly used to. Another abstraction that we are also fairly used to is little x. Let's see. So is x. So this is little x, and little x is something that we use to denote some unknown variable. So for example, we can use it in equations. So for instance, we can think of it as being an unknown number. So here we have a very simple equation, x plus 2 is equal to 7. And here x is actually standing in for 5. So we don't sort of know that a priori when we write down this equation. We can just implicitly say what x is by having it be a solution to this equation. But just to remind you that this is a bit of a weird abstraction, we'll just recall that sometimes there's actually multiple solutions to this. So here we have x squared is equal to 1. And sometimes, well, actually, there's no numbers at all that satisfy this equation. And so it is a bit of a weird abstraction, and I'm just sort of doing a little bit of X bashing to remind you that this is a bit of a weird abstraction, and we will encounter another weird abstraction in the future. But just remember that X is weird, but we're just used to it. But we can do all sorts of nice things with x. So we can solve for expressions such as f of x is equal to y. That is a standard thing that we do with x. Or we can also integrate. So f of x can be. So what sort of applications can we use for this? So let's say we have a cannon. The cannon has a certain angle x. So x is measuring this angle. Y is an observation. So we have observed the cannonball to fly distance y. So there's several different problems one can formulate with this sort of thing. One is just if we know the size of the charge and other things such as the mass of the cannonball and so on, if we're given the distance that it travels, can we solve for the angle? Okay, so that's as sort of standard physics exercise. And then another sort of question that we might want to solve is a prediction problem. So let's say we take a 10% bigger charge. So that is, there's sort of 10% more power behind this this cannon, and we fire again. Is it going to hit the castle or not? So we'll say this. The answer to this question is hit sub 10. So this is like 10% extra juice gives you what distance? Is that clear? Okay. So and let's just say that hit sub 10 is 1 if x will make the cannonball hit the castle and 0 otherwise. So if x is 0, well, it's not going to go very far. If x is sort of up at 90, it'll just go up and come down. But there's some intermediate range in which it will hit the castle. So hit 10 is 1 if x is in that range. So this is great. So if we're in this castle, we can do these physics problems and understand if we're about to get our house demolished. But it allows us to do this solution in a deterministic framework. So when I say deterministic, I mean that it's the opposite of a probabilistic framework. So what I would say that this framework is really has this as its mental model of what the world looks like. So what is this? So this is a deterministic coin flipping machine. So what you do is there's a coin, you put the coin in this little neck thing here, and then you push this armature down, and then you sort of fix it down with this switch, and then this coin, once you release the switch, this coin does a certain number of flips and lands in this bucket. And if it lands once with these settings as heads, then every time it will land heads if you put it in heads. So this is actually a figure from a scientific paper where people were analyzing whether coin flips were indeed random, or I mean, of course, they're not exactly random, but are they biased? So is a coin more likely to land heads or tails? So this is, of course, a silly view of the world. So this is not the sort of coin flipping I'm at all used to. I'm used to coin flipping that looks more like this. So this is another apparatus for flipping coins. It's called the human hand. And there's a bunch of variables that we don't actually control for. So there's this part which is called the thumb, and the thumb has a certain length. There's muscles that attach to the thumb that have a certain strength. We also, that's not really something that we usually specify when we're talking about flipping a coin. There's sort of the length of the nail, the position of the coin, and so on. So there's all these uncontrolled variables. So although this is not a random process, this is a deterministic physical process, it's still useful to think of it as a random process because there are so many uncontrolled variables. So I would say that this is a pretty good model of what a biological experiment looks like. So we have all these uncontrolled variables. we run it and we get some outcome, but it's dependent on lots of different variables, and there's certainly some noise in the measurement, so it's not perfectly reproducible. But I guess I'm being a little bit unfair. I would say if this was a biological experiment, there would also be some probability that, like, the coin would stick to the thumb and it wouldn't flip at all, or that it would flip out to outer space, or that the hand would fall off, or something like that. So there's a lot of challenges associated with biological experimentation. So given this, how do we abstract probabilistic quantities? So how do we come up with a mathematical framework that is useful for this sort of challenging situation? So it's big X. So big X is a random variable. So I'm not sure if you've heard about random variables, but they're quite a wonderful thing. They abstract the notion of a variable. So random variables don't have a fixed value. You can't just say, big X, what is your value? And it would sort of say in some hipster nasal voice, like, I don't have a value. You have to ask me for a value. So you ask this random variable for a value, and this hand flips the coin, and you get a value. So they're capricious. So they don't have a fixed value. You can't nail them down, but they are well defined. Any questions about random variables? Okay. So they're well defined, and they're typically defined by distributions. So sometimes these distributions are discrete. So there's some probability of getting a heads, and there's some probability of getting a tails. So every time we sample this random variable, we're going to get heads with this probability and tails with this probability. So it is random, but it's sort of random in a limited sense. I mean, we're not all of a sudden going to get, have tails be much more likely than heads or something like that. And we're not going to all of a sudden flip a coin and get poodle. They're sort of well-defined objects. So that's called a discrete random variable, and we can also have something called a continuous random variable. So here, instead of just deciding whether you're going to get heads or tails, you can get any value in this range, the X value range, and some of them, the values are more likely than others. So this is a relatively likely part, and it gets less likely as we go out to the tails. So what can we do with these random variables? Well, we can do a lot of the same things that we can do with little x, although they typically sit in a rather different framework. But we can solve for random variables an equation like this. So this equation says this function of x is distributed according to y, obtaining expressions such as this probability of x given this y. And so for historical reasons, I would argue, this isn't called solving this equation, it's called inference. So the mathematicians had a certain set of things that they thought were math, and then when this sort of equation came along, they said, that's not math. So now this sort of thing happens in statistics departments. So when I say solve for x, remember, big X doesn't have a single value. It has a distribution of values. So we can't just get x's value, but what we can get is we can get the distribution of values for x. So for instance, if we were flipping coins a lot, we would find out what is the probability of getting heads. And y typically is thought is sort of the data. So we can think of this f of x is distributed according to y as that there's some unknown hidden state that we would like to know about, this x. We have a bunch of data, and we have some function mapping the input states to the data. And we look for ways to solve for x. So in particular, as we get more and more data, I mean, this is secretly Bayesian inference, but as we get more and more data, hopefully our uncertainty in x is going to get smaller and smaller. So this is sort of like if we have a coin and we flip it once, and it comes up heads, and then another time and it comes up heads. Well, it might be a fair coin, or it might be biased towards heads. It might even be biased towards tails. But we don't really have that much information to say exactly what the distribution is. But if we flip that coin 10,000 times, we're going to have a pretty dang good idea of what the distribution is that determines the value of that random variable. So, any questions there? Okay. So we can also integrate with respect to x. So we integrate with respect to average out an unknown probabilistic quantity. So that's a little bit abstract. So let's go back to the canons. So here we are back with the canons. But now, instead of having one observation, we have lots of observations, so lots of different y's. We still have our x, but this time it's a big X. And it represents a distribution of what is the canon angle. So this collection of observations we think of as a distribution. So it's a distribution of these horizontal distances that are traveled by the cannonballs. And then x again, like I said, is the distribution of the canon angle. So the first problem that corresponds to the previous first problem is given an observed distribution y. So if we have a bunch of observations y, what do we know about the possible angles for x? And I would argue that this is a bit of a more realistic situation for real-world inference, where perhaps we can't measure y precisely, but we can only measure y approximately. And so it makes sense to model y as a distribution. There also might be effects such as wind. We don't know how windy it is when we're doing this. The wind may come and go. There's probably a little bit of variation in how strong the charges are, even though they're supposed to be of a fixed size. And so that we should reflect that uncertainty in an uncertainty about big X. So big X is the random variable that we're doing inferences about. So before we were solving for a single quantity x, assuming that we had one perfect measurement y, but here we have some imperfect measurements y with associated randomness. And so we're inferring what the distribution x is. Is that clear? Jeremy, how am I doing here? Jeremy? Can anyone hear me? Hello? I'm not getting any feedback here from you guys.
I think it's going well.
Is it frozen?
You just stuttered a little bit there for some reason, but you're back.
Okay.
Yeah,
can you hear us? It looks like my... Okay, now I can, but my image of you just froze, so I'll have to imagine you, and you'll have to... Oh, there, okay, that's better.
Yeah. We're just sitting very still.
Great.
Okay.
Okay, so scientific content. Is this seeming reasonable?
Yeah, I think so. Does anybody have questions they want to ask at this point?
Okay.
Good? Okay.
Okay.
Right.
So now we get to the integration. Great. So now we get to the integration part. So the answer to this first question is we do inference, and I'm not going to talk exactly about how we would do this inference, but just know that there are methods to do this solve this sort of problem. For the second one, well, assuming that we have done this inference so that we know about the probability of seeing an x given the data, what we want to do is integrate out this hit function. So this hit function, remember, is 1 if it's going to hit the castle, and 0 if not. And so now instead of a yes-no answer, yes, it's going to hit the castle, no, it's not going to hit the castle, what we can get is a probability that this 10% bigger charge is going to hit the castle. And the way we get that probability is by integrating out this hit function with respect to this thing, which is called the posterior. Okay, so why am I talking about these castles and such? Well, biological experiments are, of course, measurements with uncertainty. So instead of thinking about things like cannonballs and such, let's say that we have an individual here, and this individual has a gene, perhaps like an oncogene or something that is not good, but we're curious about what is the expression level of this gene. And let's call that expression level X. So we can do sequencing. So this is a picture of a MySeq. It's sort of a little icon for a MySeq, the latest generation of sequencers. We get a whole bunch of sequence information. That's I'm going to designate by Y. And then once we have that, we want to understand what is the risk. So what is the risk of that this person has based on the inferred expression levels of the gene. So that's where this metaphor is going. So pretty much now we're going to be talking about biology and not so much about cannonballs. But I wanted to say, so this is sort of the first part of my outline is talking about model-based statistical inference. So we can solve for X in these equations, and then we can also integrate out uncertainty to get final estimates. So here, we don't really care a priori what the angle of the cannon really is. What we really care about is if our house is going to get destroyed, if the shot is going to hit the castle, and we can integrate out the uncertainty that we have in X in order to get the probability that it's going to hit the castle. So another approach would be to find the best value for X, so sort of the most likely value for X, and then say, okay, given that, are we going to hit the castle or not? But that's, I don't think, really telling the complete story. So, okay, what is model-based statistical inference on discrete mathematical objects? So far we've been talking about an angle. That's a continuous mathematical quantity. So let's see what discrete mathematical objects are. So we're going to have a little quiz. So the first quiz question is find a number between, that's supposed to say between 2.9 and 3. So Jeremy, can you answer this difficult question?
I'll pick 2.95.
Yes! You win. Okay. So you got the full score on the first quiz question. So the nice thing about this is that real numbers form a continuum. Between any two numbers, so between 2.9 and 3, there's another number. In fact, there's uncountably infinite numbers. I'm not going to really go into that, but there's quite a lot of numbers in between 2.9 and 3. And then even once we take 2.95 and we're looking at numbers that are between 2.95 and 3, there's still a lot of numbers in between them. So they form this nice continuum. So that's kind of convenient. Here's another question. Which comes first, 5 or 6? So 5 does. So real numbers are equipped with a total order. So given any two numbers, we can always say which one comes first. So does 5 come first or 6 come first? Well, 5 is smaller than 6, so we put 5 first. So the last quiz question for this part, which I guess I'll answer because it's really tough. How far apart are 3 and 7? Well, we can take the absolute value of 7 minus 3, we get 4, and that is the distance between 3 and 7. And the take home here is that real numbers are equipped with a simply computed distance that is compatible with a total order. So that makes our life really easy if we want to do things like define what integration is. So you may or may not remember, but in the classical, so high school way of defining integration is that we have our limits a and b. You subdivide the interval between a and b into small sections. You have lots of different areas of these rectangles, and then as these rectangles become smaller, the sum of the area of the rectangles becomes a better and better approximation for this integral. So, although this sounds like pretty innocuous, all the sort of things what we're doing, like when we're taking a limit as delta x goes to 0, that's really using the structure that we had. So they have, like, we're using that a is smaller than b and that all these other numbers in between them. So that's using this total order. So we're using continuum because there's this delta x that we can shrink to 0. And we're also using the fact that this total order is compatible with the distance, which here is just the absolute value of the difference. So you're probably accustomed to this defining an integral that looks like this. You'll have to just take it on faith that you can also integrate with respect to random variables in the same way. So now, getting to this discrete thing, so what if X is a phylogenetic tree? So I imagine if you've hung out with Jeremy that you are familiar with phylogenetic trees, but briefly, there's sort of the idea is that there's some ancestral organism out here, and this ancestral organism has two lineages. One lineage splits to make these organisms, and another splits to make these organisms, and so on. So this is showing the evolutionary history of this group of organisms. So we can do really the same sorts of things that we were talking about before. We can have a little x, a big X, which is a random variable, but now instead of just being a number, it's a phylogenetic tree. We have some sequencing data. And let's say we want to understand, do these dots segregate into two different subtrees or not? So a reason why we might want to do this is actually one of the first questions I worked on when I was here at the Hutch is if you take two samples from an individual who's got a chronic infection such as HIV, does it appear that those two samples came from two separate infections or from one infection? So, for instance, if we think that it came from, if the samples look like this, so if these are the first time samples, these orange ones, and the green ones are the second time samples, this is what something might look like if you were to have a single infection. But perhaps if you have two separate infections, if perhaps somebody got HIV again in between the two testing time points, you might see a sample, something that looks like this. So this motivates us to start thinking about how can we do this sort of probabilistic reasoning when we have discrete objects, when X is a discrete object. It actually is a little bit more complicated than this because phylogenetic trees are a combination of discrete and continuous parameters, but that's, let's just say it's a discrete object. Any questions on that? Okay. So how does this actually work? So second quiz. This is a much harder quiz, and so I won't subject you to it. So what is a tree that is between T1 and T2? This is supposed to be the equivalent of what is a number in between 2.9 and 3. Well, here we have two trees, but there's really not a well-defined notion of an obvious tree that sits between these two trees. I mean, we can make up definitions, but it's not sort of God-given. So, and part of this is that phylogenetic trees have discrete topologies. So even if we found something that was between these two trees, say it's this tree T, then when we calculate a tree that's between T and this other tree, would we have another topology or not? I mean, the fact that we're doing discrete real random variable estimation is sort of a challenging thing. Phylogenetic trees also don't come with a total order. So if we're given these two trees, it's not immediately obvious which one should come first. I mean, again, you can make things up. So how far apart are two trees? There are actually ways that people have thought about doing this, and one of them is this thing called the subtree prune regraft. So where we take this part of the tree, we cut it out, and then we reattach it over here. So we can measure these distances between trees, but still, it's a little bit challenging. So it turns out that if we are given this problem, so if we're given this problem that we have tree 1, tree 2, and we want to compute this subtree prune regraft distance between them, i.e. the number of such operations that we have to do, that is an NP-hard problem, so a very difficult computational problem. So this is just to say, we can make these definitions, but sort of some obvious ones, they're not even easy to compute. So we're not going to be doing direct integration anytime soon. But we still want to do this sort of inference in the setting. We still want to be able to say what is the posterior on phylogenetic trees given a bunch of data. I'm not going to say a whole lot about this. Actually, this is about all I'm going to say. But this is something that we've been working on, that I've been working on with my postdoc, Chris Whidden. And we've made some observations about the posterior distributions of phylogenetic trees and how it can be difficult for phylogenetic algorithms to jump from one peak, so this is one section of tree space that's good, to another peak this is another section of tree space that's also good. So I'm sorry, I'm not going to go into that, but I want to get to some biology because the whole reason for computational biology is biology, in my opinion. So we're going to skip forward and go to biology, but I'd be happy to answer a question if anybody has. Okay. So microbes in the immune system. So we've already looked a little bit at microbes in the immune system. So we're interested in doing phylogenetic analysis, so inferring these sorts of random variables when we have microbes here and innate immune genes. So these actually have a fairly straightforward evolutionary process compared to what I'm about to say. It still has its own complications, but let's just say that we've checked that off. So next, the immune system. So the immune system is super weird and really wonderful and beautiful. So I want to start this off by talking about how amazing vaccines are. And this is a bunch of depictions of Jenner after developing his 1796 vaccine. So there really are not that many scientists who are pictured in this many different types of depictions. I mean, this one's like on a candy bar. It's like, this has made its way into the lore. This is not the space that scientists that usually they usually occupy. I especially like this picture because it's a picture of Ed Jenner, and you can't see his left hand. So with this hand, he's administering the vaccine. With this hand, he's actually typing on his iPhone, and he's updating his Facebook status. And he's like, "I just invented vaccination, LOL." So that's Ed Jenner for you. But more seriously, where are we 200 years later? So we've had vaccines for quite a long time. Have we made good progress in developing vaccines? So to give a little perspective on this, I could give some numbers about this recent HIV vaccine trial. It took 6 years. They enrolled 1,000 volunteers, and then out of the initial pre-screen, 16,000 made the initial cut. Out of that, they got 125 infections, and this cost $105 million. So that is a lot of work to go into just trying one vaccine. And this vaccine wasn't even very good. It was the first not bad vaccine, it seems. But this is really no way to do science. So they're expensive, slow, and it's like, they've done a good job with the moral issues, but it's still sort of tricky. So this does not lend itself to rapid vaccine development. So what would be much better would be to guide vaccine development without actually exposing people to disease. So with that, I will introduce you to something that we are all friendly with, B cells. So I can say that my mom has had B cell cancer. So even though she's had B cell cancer, I still like B cells. They're a very important part of our adaptive immune system. So B cells make antibodies. There's all sorts of complications in immunology, so I'm just going to say it once. This is a simplified view, and I will make some simplified statement. So the question really is, what can we learn from B cells without battle testing them? So without actually infecting people with a virus, what can we do to test whether a vaccine has been effective or not? So again, B cells make antibodies. Antibodies bind antigens. So antibodies are made out of two sections. This is the heavy chain here, and this is a light chain. And then the antigens, which are really just chemicals that are bound by antibodies, grab on somewhere here, or the antibody grabs onto them in this region here. And this part down here is used for signaling. Okay, so there's too many antigens to code for directly. So if we wanted to have one gene for every antigen that we would need to code for, there's no way that would happen. I mean, that would just overwhelm our genetic code. So there's no way we can have one gene here in our chromosome for one antibody. So the body has to do something tricky. So it does this. So this is just an overview, but I'm going to first talk about this top part and then the second part. So how do these things come to be? There's a bunch of these so-called germline genes. So these are different V's, D's, and J genes. And to make an antibody, you have to assemble a V, a D, and a J. So the first thing that the body does is it chooses a V, D, and a J. And then after this choice, part of each gene gets deleted, some random amount of nucleotides. So here's another. So this is supposed to represent that this part got cut out. This part also got cut out. And they're joined together with random nucleotides. And once you have this, then you can make an antibody. So this is just a diagram showing that, well, okay, this is what we're calling the genotype. And the phenotype is that it expresses this antibody. Questions? Okay. So this generates quite a lot of diversity because you're sort of multiplying the diversity of V genes times the diversity of D genes times the diversity of J genes and so on. And then there's also these random deletions and insertions. So that is the way that the body gets a whole lot of diversity. But it doesn't actually quite stop there. So there's this evolutionary process that happens. So you start with one of the product of that previous analysis. And there's these somatic hypermutations. So just there's mutations that the body puts into these cells. And the idea is that mutations that increase the affinity of this antibody for its cognate antigen are allowed to reproduce more than ones that don't. So this one here, in the schematic, it's sort of flattened out, so it doesn't fit the antigen quite as well. But these fit it progressively better. And finally, when this fits it really well, then it reproduces quite a lot. So it's this Darwinian evolutionary process. And of course, when I hear Darwinian evolutionary process, my ears prick up because that's the sort of thing that I like to analyze. So really our overall goal with this sort of work, and this is a ridiculously ambitious goal that we'll never be able to achieve, is if we have, so within each one of us is implicitly this collection of trees. So there's a bunch of different rearrangement events, and then each one has these somatic hypermutations that are accumulating. And this really, if we knew this structure, we would really know quite a lot about the history of someone's immune system. So maybe this one corresponds to the flu they got one year, or maybe this is just the flu lineage, and this was the flu they got one year, and then this is the immunization they got another year, and so on. So we would like to take a bunch of sequence data from these people, which is available, and then do inference. So we would like to be able to reconstruct what this whole history of the immune system actually was. So I'm not really going to talk so much about that goal, but I am going to talk about what happens if we want to mutate specific residues in an antibody. Is that allowed? So if we're designing a better vaccine, we have a specific antibody in mind, but the question is, will the body allow that mutation to occur? And the way that this is typically done, that you may have heard of if you've taken one of Jeremy's classes, is with the synonymous versus non-synonymous distinction. So if we have a codon, so a 3-nucleotide group, and we see a mutation that does not change the underlying amino acid, so let's say it goes from proline to proline, that is called a synonymous mutation. If it goes between amino acids, that's called the non-synonymous mutation. So the idea is that if you have lots more synonymous mutations than you have non-synonymous mutations, then that's probably a site that doesn't tolerate change very well. Because there's all these mutations accumulating, but the body throws away ones where that amino acid changes. On the other hand, if you see more non-synonymous mutations than you would think, then perhaps that's a place in a protein that really likes to change, because those changes are swept up to fixation. So so we would like to do this on antibodies, and this is really what we'd like to have at the end. Is this is sort of an antibody sequence. We would like to learn, is it under purifying selection? So is it this dominated by synonymous mutation, or is it diversifying? So does it seem like this site permits more mutation or not? So there's a certain complication in how these mutations happen. So we would like to do per site inference, but if we look at these sequences that are supposed to be evolving neutrally, we can see that there's quite a substantial difference site by site in the rate of mutation. So some mutations, some locations consistently, so these are between 3 individuals, consistently mutate more than we would expect, and some locations mutate much less than we would expect. So this is going to really make things kind of complicated. But we have a way of dealing with this. I'm sort of speeding through things, and that allows us to get an estimate of this synonymous non-synonymous ratio. So that's what we're measuring, is this. So this is sort of the scaled rate of non-synonymous up here divided by the rate of synonymous mutation. So skipping details, this is sort of what our output looks like. So if we're just looking at a single individual, we can see that some positions are somewhat neutral, so that because they're sort of hovering around 1, and some are very purifying. So this is where they have a negative, an omega, this ratio less than 1. So these are ones that don't like to change at all. So it does appear that there are some that do think that change is okay, but I would say it's dominated by purifying selection. So this is just a histogram telling a somewhat same story. There's a bit of a difference here that I can tell if you want to ask questions. But this is the number of genes that that were identified as being under purifying selection versus being under neutral, which is gray, or diversifying selection. So one of the things that really jumped out at us is, what the heck is going on down here? So these are ones that appear to be very under a lot of purifying selection. So we can see that a lot of the genes are under purifying selection because this is a high bar, and and the coefficients of selection are quite small. So you can map this onto a sequence. So here, this is sort of a blow up of an antibody. So an antibody looks like this. So if we just magnify this part, so we're mostly looking at this blue segment, this part of the heavy chain. So that is most of what you're seeing here. There's a little green stick figure that's representing this green light chain. So this is the light chain here. And that residue that we were looking at just now is this residue right here. And the reason why it makes sense that this residue would be under really strong purifying selection is because it sits in the middle of the heavy chain. And in order for the antibody to maintain its integrity, in order for it to be a folded protein, it needs to have this ring not change, because that ring probably makes connections with lots of other things that are buried inside of this heavy chain. And in fact, so this is the same color scheme that orange is purifying selection and blue is diversifying selection, and overall, statistically, the ones that are sort of touching the light chain or buried inside are ones that tend to have lots of purifying selection, again, because, in the story at least, is we think that it is because those stabilize the heavy chain, but then there are others, such as this one, that are sort of sticking out, so it's allowed to diversify. So I'm really just going to stop there. I think it's about time. But I would say that physics to calculus is as computational biology is to solving for and integrate with respect to random variables X. So this calculus of random variables is really a super important, wonderful thing. And this is called model-based statistical inference. And it happens in statistics departments and now somewhat in computer science departments. And we can do this when random variables are discrete, such as phylogenetic trees. And in kind of a non sequitur, antibodies are awesome, and we can learn about natural selection on them using this framework. So with that, I will just say thanks to my collaborators. Trevor Bedford is quite a clever flu phylogenetics person. At the Hutch, Connor was in my group. He was a programmer, and now he's at Google. Vladimir Minin is a statistician at the University of Washington, and then Chris Whidden is in my group. So thank you for your attention, and thanks for the funding as well.
Thanks very much, Eric. We have time for questions if folks have them. So most of the students who come to this come from a biology background. Do you have any advice for biologists who are interested in getting involved in or thinking about the possibilities, sort of career possibilities in computational biology? What sort of the best way to learn about these sorts of approaches might be?
Yeah, I mean, taking university courses is great. I had to learn all this by myself, so I've really never taken a statistics course. And I took one computer programming course. But you're at a university. I mean, this is a great time to learn things such as computer programming and statistics. So, I guess my main advice would be that the introductory parts are really boring. I'm sorry, like t-tests are boring, and writing a hello world program is boring. But once you sort of get through the boring parts, there's some really exciting rewards at the end.
What's the sort of mix of computational people and empirical people at a place like at the Hutch?
Oh, I am way out in the tails. Basically, there's a computational biology program, and there's one other person who has an all-dry lab. Well, kind of two. So, and then the other computational biology people have wet labs. One actually has a business that does wet lab work as well as analysis. But yeah, there's if you want to work at a place like the Hutch, a much better idea is to get a biology degree.
So, questions about the research platform?
I have a quick question. Where do you get the sequence data for these last analyses you just presented? Are they from other labs at the Hutch, or?
Yeah, we collaborate like crazy. I mean, I've helped biologists design experiments, but we pretty much just have a huge number of collaborations going. And I think it's really fun because on one hand, sometimes the questions that people have are really easy. And then hopefully you can just sort of knock them off and go off and do something else and help them a lot. And then that's great. And then sometimes they're very interesting and very difficult. And that's when I get really excited. And we get to write a grant and think about how we might solve a whole class of problems. So I mean, yeah, I mean this grant here is all about antibody stuff, and the reason why we got that is because we were working with this HIV lab and they were doing HIV antibody sequencing, and then yeah, the project certainly took a life of its own.
May I ask a question about metagenomics, actually?
Great.
Yeah, I just want to know, for most assemblers available now, they always try to make different contexts into one context, try to do that. But for the metagenomics, we want to assemble different contexts, but at the same time, we want to separate them because you have different organisms in there. So what kind of our algorithm involved in that?
Yeah, that is a great question. So if I understand the question correctly, you're saying that most assemblers just really try to glue together as many bits of sequence as are possible.
Right.
And irrespective of this sort of makes sense biologically or not. So I don't actually work on metagenomic assembly. There are a number of people who do. But I guess just in the context of this talk, I would say that this shows how a sort of pure heuristic, which is assemble as much as possible, is not as good as some method that explicitly tries to model both the generation of the sequencing and what organisms in a metagenomic sample look like. So I think that this sort of shows that a heuristic approach is a little bit more limited. That said, it's very difficult to write a fully probabilistic genome assembler. But people do have sort of approximate ones. Yeah, does that answer your question? Maybe not.
Yeah, sort of.
I mean, I don't work on metagenomic assembly, so I can't quite speak to that exact area.
Okay, thanks.
Any other questions? Okay, well, let's thank Erick one more time. Yeah, great job.
Thanks.